Greetings. This video is about B19 Parvovirus causing fifth disease in children. B19 Parvovirus was discovered in the year 1974 and it belongs to Parvoviridae family. Among different groups of parvoviruses, B19 causes infection in humans. It is highly contagious and spreads from person to person and often results in very mild symptoms. B19 Parvovirus causes fifth disease, that is slab cheek appearance. This is common in school-aged children. The structure of B19 Parvovirus composed of linear single-stranded DNA. It is a non-enveloped virus. The size of B19 Parvovirus ranges from approximately 22 to 24 nanometer in diameter. Mature infectious viral particles have a molecular weight of about 5.6 into 10 to the power of 6 kilodaltons. B19 Parvovirus has a single non-structural protein NS1 on the left side and two structural proteins VP1 and VP2 on the right side of the genome. Transcription process of B19 Parvovirus produces at least 9 mRNA transcripts. Here is a structure of B19 Parvovirus made of VP1 minor capsid protein, single stranded DNA linear genome and VP2 major capsid protein. Modes of transmission of B19 Parvovirus. B19 Parvovirus is present in saliva. It can spread through airborne droplets when an infected person coughs or sneezes. It can also spread through blood. Pregnant women with parvovirus can spread the virus to the fetus through placenta, that is vertical mode of transmission. Replication of B19 Parvovirus. In nature, B19 Parvovirus mainly infects human erythroid progenitor cells from bone marrow and liver. B19 virus DNA replication was first observed in suspension culture of human erythroid bone marrow from patients with hemolytic anemia. Parvovirus first enter into the cell and attachment to the host receptor initiates endocytosis of the virion into the host cell. The virion consequently penetrates into the cytoplasm and reaches the nucleus where uncoating occurs. In order to replicate, viruses have to recruit an origin of replication. The non-structural protein NS1 recognizes the replication origin. Single copy of DNA is initially converted to double-stranded DNA. Transcription of newly formed double-stranded DNA give rise to viral mRNAs and are subsequently translated to produce viral proteins. Newly synthesized single-stranded DNA can either be converted to double-stranded DNA and serve once again as a template for replication, or it can be encapsulated to form new virions that are released by cell lysis. Pathogenesis of B19 Parvovirus. B19 Parvovirus binds to host cell receptors in the respiratory tract and enters the cell. It then translocates its genome to the host nucleus where replication and assembly of the virus occurs. Finally, the cell lyses and releases the mature virions. Viremia occurs and creates symptoms. The IgM antibodies appear when viremia resolves, which is approximately 8 to 10 days post-inoculation. During the viremic period, reticulocytopenia occurs for 7 to 10 days. 1 week after the appearance of IgM antibodies, the IgG antibodies appear and coincides with the appearance of rashes and arthralgia. The P antigen is the cellular receptor for B19 parvovirus that causes erythema infections in children. Fifth disease is the clinical disease caused by B19 parvovirus in children. One of the most common presentation of B19 parvovirus infection is fifth disease. Fifth disease is typically recognized by the slapped cheek rash. It occurs most often in young children. Fifth disease got its name because it was fifth in the list of historical classifications of common skin rash illness in children. This picture shows the slapped cheek appearance in a school-aged children caused by B19 parvovirus. Symptoms of B19 parvovirus infection are usually mild and may include fever, headache, sore throat, cough, rashes, painful or swollen joints. Laboratory diagnosis: enzyme-linked immunosorbent assays for the diagnosis of B19 IgM and IgG antibodies. The PCR is a very sensitive method for detecting B19 parvovirus in clinical samples. Some of the treatments for the infection caused by B19 parvovirus include intravenous immunoglobulin therapy, acetaminophen, ibuprofen, and anemia patients receive blood transfusion. Prevention: no vaccine available. Improved personal hygiene by washing hands often with soap and water and avoid contact with people who are sick. Thank you.